Let's do this question here. This one is extra hard. So I know it requires some understanding of a different module, but I think the question gives us enough. So I'll just break this down for us first, and hopefully we can understand the question. So the rabies virus is a single-stranded RNA virus. It contains and codes for only 5 proteins. The diagram shows the structure and reproduction of the virus. So what diagram 1 is showing is just what the virus has. So a virus is just a collection of proteins with some kind of viral RNA inside, right? So it says it contains LP protein, which is RNA polymerase, contains some M, G, and N protein. Doesn't say what they do. We'll just assume they're part of the structure. And it contains a strand of viral RNA inside, right? So that's what our virus is made of. And before I show you this diagram, just a bit of background information. Viruses, if you might remember from last year when we did diseases, viruses, they're like a bit like terrorists. They hijack a cell and then they use its like resources to make more of themselves, right? But you can kind of figure that out from this question anyways. Yeah, Lucas, what's up?
In the first diagram, is it pointing to where the protein's like stored, like where the code for it is stored, or what it's made of?
What it's made of. Yeah, what it's made of. Yep. So what do we see in this diagram here? Well, I'll look at the question first. It's asking us for, it says the RNA gets transcribed, translated, and replicated in the cytoplasm of host cells, right? So it's 5 marks, and we have to use the diagrams to explain the role of RNA polymerase in the reproduction of the virus. So what I have to do here is I have to figure out how I can see all the processes of transcription, translation, and replication here, and we can link that to how this leads to the creation of more offspring or more viruses at the end, right? So just by looking at my diagram, what are some things I can immediately notice? Well, I can see here that it's talking about translation, right? So this is where translation's happening. I'll go back to that first, but because here we get mRNA after our viral RNA gets read through RNA polymerase, that sounds a bit like transcription, right? Going from RNA to mRNA, right? I know it's a bit different because in I guess for us, it's DNA to mRNA after being read by RNA polymerase, but because, you know, RNA polymerase is happening here, this is where transcription is occurring. So I'm just going to label this as transcription. Right. This is translation, right? Because our mRNA is being, I guess, it is skipping a few steps. I mean, there probably is the ribosome working, and then there's complementary tRNA, and that's eventually making our proteins, right? So that's translation without the steps, and then it makes these proteins. Right now, these letters might seem a bit random to you, right? L, P, M, G, M, N. But these are all the proteins that are in diagram 1, right? That are in the structure of the virus. So we can see here that through translation, it's building all the necessary proteins that are part of the virus that's being replicated here, right? So that's transcription, translation. The last one is replication, right? Where is replication happening? It's happening in this side of the diagram here, right? So this part here is replication. Right? Right? Now, we know that in order for something to replicate, we need polymerase, right? And how convenient, right? It says these 2 proteins are what get us from viral RNA to complementary viral RNA. And then these 2 proteins, again, make more copies of our viral RNA again, right? So that tells us that these are probably polymerase, and that's confirmed when I read the diagram 1 here, right? It says L and P protein are RNA polymerase, right? So that means that these are probably also responsible for making more copies of my viral RNA, right? So in this case, what's happening here is for DNA replication, we have our double-stranded DNA, right? And then we know that it's semi-conservative, right? So one strand is new. It looks a bit like this, right? But our RNA is single-stranded, so it's a bit weird because, you know, we can't just make an exact copy of this strand, right? But that's what we want to make. We want to make a copy of this strand. So to do so, we make a complementary copy, right? And then from that complementary copy, we make back our original strand that we actually want, right? So I'll just draw that again to make it more clear. So let's say I really want this red strand, right, on the bottom. But I can't just get one red strand and make another one just like that, because how a polymerase works is it has to attach complementary nucleotides, right? So what happens is I start off with my red strand here, my viral RNA, right? I shouldn't draw like that, right? So my red strand here, and then it makes a complementary strand, right? So if this is my vRNA, it makes a complementary strand that is associated with it here, which I'll draw as black, right? But we don't really want this strand here. We want the red strand, right? So we get polymerase to read the complementary strand, right? To read this thing here so that we get our original red strand back, right? Which is where all these copies come from, right? Does that make sense? Kind of, right? So that's the whole thing, right? So part of the question, well, it's 5 marks. Yeah, and I did say it was very hard. So we have to explain the role of RNA polymerase in the reproduction of the virus, right? So we have to talk about all 3 processes of transcription, translation, and replication. So that's here, right? So what I'll do is I'll just chunk it for us, kind of. Right, replication, right? So I need to refer to my diagram here, right? So I should probably say, like, RNA polymerase is the LNP protein, right? And I can talk about how this creates creates complementary RNA strands. And then I say this strand, so complementary RNA is read again to produce many copies of viral RNA. Right? Cool. Now, secondly, I'm gonna put this into 3 sections, so not like 5, but hopefully you can kind of see how I get the 5 marks. So the next part I'll talk about is transcription. Right, in this case, I can say RNA polymerase transcribes viral RNA into viral mRNA, right? And that's transcription, right? Then I can talk about translation, right? So here is where I can again talk about my diagram, right? So this or these mRNA sections code for was it L, P, M, N, was it G? Was it G? I don't remember. G proteins, which are important for virus assembly. Plus, I'll also link it back to replication because we know we use the LNP proteins as a polymerase to replicate our strand. So plus replication. I might also say that combining all these strands together gets us our, I might just say therefore we create many copies from one virus. All right, cool.
Do you need to say like code for LPMN2 proteins in diagram?
I don't think you need to mention diagram too explicitly, but like just the fact that I've named the proteins tells the marker that I like actually looked at the diagram. I'm not like just saying stuff out of like my butt, which is why you should probably mention the name of the proteins, like the letter. Otherwise, you're not showing the marker you're engaging with the stimulus.